Yeah. Good evening, everyone. Welcome to today's Cell Talk webinar. Today, our speaker Vijay Paudul will be delivering the topic on human longevity and Alzheimer's disease variants Act Viva, Microglia, and Oligodendrocyte gene networks. So, Vijay Paudul is working as a scientist bioinformatics at Renex Life Science. At Renex, he is leading multiple research projects on bioinformatics, genomics, and next generation sequencing research related activities in the organization. His current research projects include the development of personalized neoantigen cancer vaccines and the utilization of publicly available multi-omics cancer datasets to create new insights into cancer biology and its application in cancer therapy. Vijay Podu has completed his PhD in Life Sciences from TMC ACT RAC, Omi Baba National Institute, Navi Mumbai. His PhD study focused on cancer genomics involving multi-omics analysis of Gallium brain tumor subtypes. Vijay has over 10 years of experience in the wet lab as well as in silico approaches for multi-omic studies, which include exome sequencing, whole genome sequencing, transcriptome sequencing, and analysis. Previously, Vijay has worked at various graduate and postgraduate degree colleges as an assistant professor. Thus, he brings multi-omics expertise and teaching experience to the Omics 5 team. At Renix, Vijay has established bioinformatics pipelines for personalized cancer genomic analysis, and his research has highlighted the importance of molecular marker analysis in personalized cancer therapeutics to match the treatment strategies to patients' cancer tissue molecular characteristics. His contributions include assessment of patient HLA genotype status for tailoring the personalized neoantigen peptide therapy and glioblastoma subtype analysis. His larger aim is to utilize personalized genomic approaches for aiding effective personalized treatment strategies for cancer patients. So I welcome Vijay to start the presentation.
Thank you, Chaya, for the introduction. For today's Cell Talk Journal Club activity, I have selected a paper, Human Longevity and Alzheimer's Disease Variants Act via Microglia and Oligodendrocytes Gene Networks. The original authors are Andrew C. Graham et al. The title for the presentation is Novel Genes Identified with Possible Role in Human Longevity and Alzheimer's Disease. The topic selected was Omics and Longevity. The journal it was published is titled Brain. It has an impact factor of 11.9 and it was published in year 2025. This research has impact on aging research, human longevity research, and Alzheimer's research. So coming to introduction, as we have seen in our previous presentations, the aging, it results in the decline in the function of an individual. So the aging, it is defined as the declining of the adult tissue function after development. That is, once the person reaches adulthood and his body development is complete, after that there is results in the decline in the functioning of the adult tissue, and that is what we call as biological aging. So aging, it results in decline in central nervous system functions due to cellular changes in the different brain regions, and that's why we see different kinds of functional impairments in the older people. These include impaired spatial memory, like forgetting where they put the things, where they are, or forgetting routes to their home or park, etc. And there is also impaired episodic memory, having trouble remembering when certain things happen. And there is also impaired executive function because of these impairments. So therefore, older people, they encounter problems in their day-to-day activities due to their impaired memory functions and central nervous system functions. So aging is considered as a primary risk factor for the number of neurodegenerative conditions such as Alzheimer's disease. So many degenerative diseases that occur in the older population, such as Alzheimer's disease and Parkinson's disease, these are specifically occurring in the older age. Coming to the general background. So myelin, it is a layered lipid-rich membrane which is produced by oligodendrocytes and that ensheathes the neuronal axons. So it is a protective covering around the neurons, which provides metabolic support to the neurons for its proper functioning, and also it enables fast propagation of action potentials. So this myelin, it is a repeat rich, therefore it functions as a barrier to the conduction, and which protects the conduction of signals into the action potentials in the neurons. So there is also another kind of cell which is important to the brain function, that is called as microglia, and they also serve as a protective role in the remyelination process. And it is these cells have immune functions in the brain. So microglia, they by their phagocytic action, they clear the degenerated myelins that accumulates during the aging process of the brain, and also they phagocytize different metabolic products that are excreted out by these cells, and kind of detoxify the brain cells and the brain intercellular regions. They also secrete growth factors and cytokines that support the oligodendrocyte maturation, and also they are helpful in the remodeling of the intracellular matrix. So along with these cells, there are different types of cells that get affected due to the aging process, and these include neurons, macrophages, microglia, and other supportive vasculature that occur in the brain tissue. So it has been found that myelin disruption with aging in rodents has been shown to be driving accumulation of amyloid plaques that are associated with the Alzheimer's disease pathogenesis. So as we can see, the impaired brain function and aging, it is correlated with the impaired function of the cells that are specifically occurring into the central nervous system, such as myelin, microglia, and neurons. So present challenges include that what exact molecular mechanisms are affecting the aging and Alzheimer disease, and also how these mechanisms are affecting the brain function. These are not well understood. And it is also not clear which of the many altered pathways and genes and their functions that have been yet identified, how they are affecting or driving the brain aging. So in order to identify that, this paper has an objective for this research, that is to gain better understanding of the age-dependent processes within the CNS that contribute to the lifespan and Alzheimer's disease pathogenesis. Once we understand which age-dependent processes are driving the aging phenomenon, or also protecting the aging within the CNS region, then we can identify those particular genes and pathways, and then we can use those genes and pathways as target to prevent age-dependent CNS disorders, and also age-dependent decline in the brain function. So coming to methodology, so this research paper, adopted a three-step approach to identify significant biological pathways that are dysregulated with age and which are enriched for genes associated with longevity or Alzheimer's disease risk. So in the first step, they identified the common human genetic variations that are commonly associated with aging and Alzheimer's disease. For that, they utilize different data sets that have been published by different research groups, and they combined those data sets and identified some common genes that are involved in the aging. And for that, they aggregated the different single nucleotide polymorphisms that have been yet associated with aging. And those single nucleotide polymorphisms, they tested for one particular genes, and during that they accounted for linkage disequilibrium, which might contribute to the disruption of the association of the gene for the phenotype because of the recombination events, and also they accounted for number of SNPs per gene. So some genes might be long, so they might have more SNPs as compared to short genes, which will have smaller SNPs because of the smaller nucleotides that are contained in that particular genes. So all these things were accounted in this particular step. So first, then when we identify those genes, in the second step, they assess the enrichments of those genes in the age-related gene co-expression networks. So what genes are actually co-expressing with each other? So these were analyzed, and where those particular genes are occurring in these association networks, that was analyzed. For that, they used bulk transcriptomic profiling of the human mouse hippocampus, and at the next level, they use single cell RNA sequencing, which is a sequencing of each cells in the tissue as compared to the bulk transcriptomic profiling, and in that they use mouse hippocampal microglial region. In the third step, they performed transcriptome-wide association study, and this was done to identify and confirm new risk genes associated with longevity, and compared these results with the genes associated with Alzheimer's disease in the recently published Alzheimer's disease transcriptome-wide association studies, so as to validate their findings. So this is the representation of the three-step methodology. As we can see in the first step, they are performing the analysis of different GWAS, gene-based variants, which were found to be statistically significantly associated with human longevity and Alzheimer's disease. And in the next step, they perform gene expression co-expression analysis for the RNA-seq data in the bulk hippocampal region and single cell in the hippocampal region. This was done in human GTX data as well as C57BL6J aging mice in the bulk hippocampus and also single cell hippocampus. And by that analysis, they found different genes that were part of the enriched network modules, and in which those particular genes were found to be statistically significant in co-expression analysis. Then in the next step, the significant genes that were identified in the earlier step, they use those genes to find their association with longevity and Alzheimer's disease. For that, they use significant enrichment in co-expression network analysis and also transcriptome-wide association study analysis. So coming to results, in the co-expression analysis, they found 17 gene modules whose expression was significantly correlated with age in human hippocampus. So these different modules, these different 17 modules, they are identified by different colors. So these are different gene ontology modules, and these are grouped into three different groups, that is synaptic groups, cell functions groups, and glial groups. So these gene ontologies, these were found to be statistically significant. Where these 3 stars, it means that the p-value is statistically significant as less than 0.0111. So once they identified those modules, they used those modules for gene co-expression analysis in the human hippocampus. In the first part, they identified the 152 genes in the human microglial network and 150 genes from human oligodendrocytic network. Both the genes, as we have seen, in the important for the proper functioning of the brain. And in that, they found 152 genes, and out of that, these genes depicted in red, these are the hub genes, which are connecting the rest of the genes. And in this 150 genes that were found in oligodendrocytic network, again, the hub genes are shown in the red. So these are the significant genes which are found to be enriched in the human oligodendrocyte and human microglia. Again, in co-expression analysis, they did an analysis called as preservation of the modules, identification of preservation of modules. So they did this in 2 different ways. That is the preservation of human GTX hippocampus derived modules in the mouse data, and in the second part, they tried to identify which particular modules are from the mouse are preserved in the human hippocampus data. So in that case, they identified a threshold in which human oligodendrocytic and human microglial modules, these were found to be preserved in the mouse data, and in human hippocampus data, the mouse, the microglial module was found to be significantly preserved. In the next step, they performed the co-expression analysis of the modules. So in that case, they found 8 modules which were found to be significantly showing co-expression with these different cell subtypes. So this ARM associated module, it had shown, it is showing highly significant association with age. That means this module is associated with aging related decline, may be associated with aging related decline in the brain function. And these are the different modules, that is homeostatic microglial 1, homeostatic microglia 2, transitional microglia, activated response microglia, and IRM, that is interferon responsive microglia. So these were found to be associated with these different modules and these traits at statistically significant levels. Then they performed a pseudotime analysis, and in that pseudotime analysis, they found a bifurcating trajectory. So this is a bifurcating trajectory in which these different cells are arranging in a bifurcated manner. So in that case, they find different clusters that are showing amyloid responsive module and interferon responsive module as to be significantly enriched. And here in this, it is ARM associated module expression along with activation trajectory, which is bifurcating. And in that case, we can say that these amyloid responsive genes, these are getting highly expressed. And here in this part, interferon responsive genes are also getting highly expressed. They also compared the gene expression of ARM associated module in aged microglia. So in adult mice of 6 to 8 weeks and aged mice of 16 to 18 months, in that case, they found that there is increased expression of these ARM associated module in the aged microglial cells, implicating their role in the aging. So here we also see the same pattern in interferon module expression. So here also, if we see the aged mice, there the expression is significantly increased. But in HM2 associated module, we see that the expression is reduced in the aged mice. Here, again, in a gene co-expression network analysis from microglia, they analyzed single-cell RNA sequencing data, and they found these different genes in mice gene expression networks to be significantly expressed. So in ARM-associated modules, SPP1, LILRB4A, CLEC7A were found to be the hub genes. In interferon modules, IFIT1, 2, and 3 were found to be the hub genes. Same in HM2, these were found to be the hub genes, and in the phagolysosomal module, these were found to be the hub genes. They also analyzed this module preservation in the data that was previously published by Nugent et al. And they also found there that these same modules, that is HMT associated module, interferon modules, ARM associated modules, and phagolysosomal modules, TGF beta modules, these were found to be significantly preserved in that analysis also. Then they again analyzed the effect of these modules expression in the demyelination process. So they performed this kind of analysis where they fed young mice in a demyelinating diet, that is induced by cuprizone. It is a chemical that induces demyelinations of the neurons, and that results in the mimicking of the aging phenotype into those neurons. So what is the effect of these myelination? It was studied, and they found that this ARM associated module, its expression increased in the mice which were fed with cuprizone diets, which result into demyelinations. So as demyelination increased, so this gene expression also increased. Now what this implicates is that this demyelination is the process in the which is an aging related process. So when individuals ages, there is found to be increased demyelination in the aging population, and therefore if we see that this particular module is getting increased, then there is a implication that this particular module is significant in the demyelination process and also has an impact on the aging related decline in functioning of the brain. When they studied the interferon module, initially at 5 weeks interval, it showed increased in the expression of this module, but after that, after 12 weeks, it showed reduction. The same can be seen here. In the first, after 5 weeks, it showed increased, but after 12 weeks, it showed decrease. HM2 associated module, it showed decreased on the cuprizone diet after 5 weeks and also after 12 weeks. Same with HM1 associated modules, it showed decrease after 5 weeks and also after 12 weeks. And in TGF beta module, it also showed decrease after 5 weeks and remains kind of stable after 12 weeks. After that, they performed transcriptome-wide association studies in which overexpressed or underexpressed genes that are represented by this Z score, these were found to be significantly associated with the aging phenotype. So here in that longevity genes that are from the whole blood, they found that FOXO3, which is routinely implicated in the aging and Alzheimer's disease, was found to be significantly associated. Together with that, IRAB2 and PSMA4 were significantly associated. Then in CD14 plus monocytes, they found IRAB2 gene. In longevity genes in the cortex, They found CASP8 and CDK10 and other genes. And in hippocampal region, they found FLACC1, HPR, and AR17A and other genes to be significantly associated. Coming to discussion, so genetic variations associated with Alzheimer's disease enriched with age-dependent oligodendrocytes and microglia. So this is significant because immune system has been recently implicated to be an important factor that is preventive of aging. And oligodendrocytes and specifically microglia, these are providing protective function into the brain. Oligodendrocyte providing physical and physiological protective function to the neuron, and microglia, they are serving as an immune role to clear up the brain from toxins and also from pathogens. In this study, new genes not previously reported to be associated with Alzheimer's disease were shown to be possibly having a role in age-related responses. And in the recent JIVA study, they also showed genes such as ANKH, GRN, PLECA1, SNX1, UNC5CL as new genes of interest. So these got replicated in the recent study, and therefore this validates the analysis that is presented in this particular study. And variations associated with longevity, these are enriched in a single cell homeostatic microglial network, and homeostatic functions of innate immune cells might be a significant contributor to longevity. Again, highlighting the significance of the immune cells and immune health into the longevity and preventing of degenerative diseases. This study also has multiple limitations, and first major limitation is that this entire analysis that is presented in this paper, or bulk of this analysis, is bioinformatics analysis, and this analysis requires further wet lab validation supported by wet lab experimental results. The second limitation is that the longevity, it is a property of whole organism and not just the brain. Whereas Alzheimer's disease, it is a phenotype that is resulting into the brain degeneration due to the aging. Now, there are different systems that are responsible for longevity also, that is vascular system health, excretionary system health, immune system health, etc. So this cannot be seen in isolation, but the impact of these study outcomes, these are applicable to the brain health and Alzheimer's disease, but it doesn't have in totality effect on the longevity, because other systems are also responsible in the outcome of longevity. The third limitation is that the gene level analysis of GWAS data is restricted to gene boundaries that can miss some non-coding variation associated with genes of interest. The GWAS data, this does not include the entire genome, and also whatever genes that have been selected, there might be some surrounding regions where some non-coding regions might harbor some SNPs which are not part of this particular study at all. So therefore, those SNPs may be missed. Then fourth limitation is that this gene-based analysis identifies the significance for each gene without accounting for linkage disequilibrium between SNPs in other genes. Linkage disequilibrium, it is a phenomenon which results due to the recombination, genetic recombinations during the meiotic process of the formation of gametes. So due to that, these associated gene SNPs, they might get segregated. So this is one limitation, that this linkage disequilibrium between these genes and SNPs, it should be accounted. Fifth limitation is that this analysis of mouse single-cell RNA-seq data potentially missed some age-dependent Alzheimer's disease-associated gene modules that might be detected in human data. So human genome and mouse genome have some differences. There are some similarities, that's why we use it for experimental system, but these mouse results might not be completely applicable to the human data. The sixth limitation is that use of monocyte data as a proxy for microglial gene expression might be imprecise, because there are some transcriptional and phenotypic differences between monocyte-derived brain macrophages and microglia. As they have used blood for this analysis also, so microglia being the kind of functioning similar to monocyte in the brain. So therefore, though they are functioning similarly in different systems, but they are not exactly same thing. So therefore, these transcriptional signal differences will be there, and one thing cannot be replaced for the other. Seventh limitation is that the current pace of GWAS is fast, and new GWAS studies are coming recently. So this analysis and the comparison in the study it will be available soon. That means whatever findings they have presented in this particular study, they might be replicated or repeated in some other study that might come with improved Jiva strategy that will come into the research next publication. So most Jiva's data sets, they are from Caucasian individuals, and the gene variants identified might be dependent on ethnicity. So this is a major issue that many data sets in the advanced countries, they are mostly comprised of the Caucasian individuals, that is white individuals, and African Americans, Asians, um, peoples of Asian origin and other ethnicities, their data is not very common in those particular data sets. So whatever findings that are being presented here, they might not be universal, and they might be mostly applicable to the Caucasian individuals and not to the people of other ethnicity. Coming to conclusion, so these co-expression networks in aging brain that are enriched for genetic variation associated with Alzheimer's disease or longevity, these were identified in this study. Genetic risk for Alzheimer's disease is enriched in age-dependent activated microglia and oligodendrocytes. Genetic variation associated with longevity is enriched in homeostatic microglia. And then next, the genetic variants associated with longevity and Alzheimer's disease both fall on microglia but control different and opposing states of their function, that is homeostatic function versus activated function. So in total, the immune related functioning of microglia and its activity and associated modules, those have been found to be significantly enriched in this analysis, which comprised of genome associated study data and also gene expression data from these cells from mouse and human brain tissues. This research has an impact on understanding the function of priority risks and hub genes that might allow to develop interventions to slow age-related brain decline and Alzheimer's disease. So whatever genes and that have been newly identified and that have been previously identified, if these are further studied, then we might have some intervention strategies to prevent age-related decline and also Alzheimer's disease. These genetic networks that drive aging, Alzheimer's disease, and inflammation might help to predict the physiological age of the brain and Alzheimer's disease progression more accurately in individual people by generation of biomarker and genotyping assays, which is currently missing in the medical field. So once we get those metrics to identify how much brain aging is there and how much of those neurodegenerative diseases are progressed, then proper interventional strategies or preventional strategies can be suggested to the patients. Then these findings might allow earlier tracking of disease progression and may provide new targets for Alzheimer's disease prevention and facilitate further work into healthy CNS aging. So coming to the conclusion, thank you for patiently listening.
Thank you, Dr. Vijay, for your detailed talk. Now floor is open for audience questions. So I don't see any further questions or any questions from audience. So please move to the last slide. Yeah, before conclude this webinar, I would like to inform you all that next presentation will be given by Uma Rakshita on dietary protein intake in midlife in relation to healthy aging results from the prospective nurses health study cohort. Thank you for your kind attention for joining today's webinar. We appreciate your time and engagement. We hope to see you in the next webinar. Thank you.